Hi, my name is Cusihuallpa, and this is my second time applying to REPUBio and the last, I hope. But bueno, this year I had a chance to intern at the Laboratory of Reproductive Physiology at Universidad Nacional Mayor de San Marcos. And during my stay, I worked on two research projects related to oocyte and embryo quality in alpacas. But today, I want to tell you the story of the one that challenged me the most, academically, technically, and personally. So let's start. Well, I worked under the guidance of my mentor and now my friend MSc Juana Quispe on a project that aims to identify molecular markers of oocyte competences and embryonic development using single-cell transcriptomics. And that's because in alpacas, um, the in vitro fertilization success rates are still very low. And while techniques like brilliant resil blue staining help classify oocytes by quality, they can alter the ooplasm and affect embryo development. And that's why this project focuses on a better approach, which is analyze gene expression directly through mRNAs. So my journey in this project started with something simple, maybe, but delicate, that is recovering oocytes. So for that, we received alpaca ovaries from Huancavelica after 22 hours of transport by road. From there, I aspirated the follicles and then isolated cumulus oocyte complexes and classified them by competence using BCB staining. With this stain, when we see the oocytes under the microsteloscope, the oocytes that turn blue were considered high quality because they have lower G6PD activity and higher chances of developing into embryos. But in contrast, the ones that stay Reyes were likely low competence and less likely to reach the blastocyst stage. So then came the critical part, that is the molecular part. So for that, we need to denuding the oocytes, extracting the mRNAs with magnetic beads, and performing complementary DNA synthesis. Honestly, this was the most intense part for me because I had never done anything like that before. I mean, I didn't even know what cDNA really was when I started. Uh, I was like being thrown into deep water, but it made the learning unforgettable. I mean, uh, mm, was rare. But well, we also perform in vitro modulation to get M2 oocytes and apply the same process, but we still have to proceed with the embryo samples. By now, our results are high quality cDNA from GB and M2 oocytes ready for sequencing. And now, what I learned of that experience? Well, throughout the internship, I learned much more than lab techniques like RNA extraction, staining, or travel hosting. I learned how research works in real time with its complications, surprises, and problem solving because exist. For example, in my lab, there were very few oocytes and multiple projects needed them. At one point, there were even tensions in the lab, but thanks to honest conversation between our mentors and team and my PI, Dr. Balta Valdivia, we found ways to collaborate fairly. On top of that, where delays affected sample delivery, and for me personally, adapting to Lima, to long days in the lab, and to the pressure of working in a new file was intense. But I keep asking questions, learning from mistakes until now, and trusting my team. So this project pushed me out of my comfort zone, but helped me grow in every direction. I think. And today I feel more confident, more skilled, and more determined to keep contributing to science and obviously to the future of research in Peru. So that's all. Thank you so much for listening to my experience. I hope to continue drawing with REPUBio this time and maybe even inspire someone else along the way. So, bye.